The single most important thing that a cell needs to do as part of the cell cycle is to replicate its DNA. Now, this process of replication has to be quick and it has to be faithful. It has to keep the integrity of that genetic message intact. So the reason it has to be quick is that some cells undergo cell division all the time. If you're a bacteria, you have the ability to replicate once every 24 hours. Now, if DNA replication takes too long, it's going to slow that down. Many of the cells in our body don't replicate that quickly. Some things like our epithelial cells that line our stomach maybe do that once a week. So DNA replication doesn't need to be quite as fast, but it still needs to be quick because besides the fact that there are other things that have to happen, the cell has to continue functioning in its normal environment. And if it's spending too much time and energy replicating its DNA, it's going to be a problem. More important than quickness is fidelity. So the cell needs to maintain the integrity of its genetic message for really only one reason, so that the next generation of cells has the correct information to continue growing and developing. If you're a skin cell and you pass on the incorrect information to the next generation, that cell is not going to be able to function correctly in its given situation. So even though all cells contain all genetic information and not all information is used in any single cell, all cells have to maintain everything and pass it on correctly. Another reason that fidelity is important is because even if you are not producing the same type of cell, let's say that you are producing a sperm cell that is going to fuse with an egg and produce an individual. You need to make sure that the information you're passing on to the next generation of offspring is also correct, or your offspring is going to have problems. So maintaining the genetic information, maintaining the DNA without errors, replicating it without errors is really important to the next generation of cells, whether that next generation is the same type of cell or another new organism. So before cell division can go, DNA has to be replicated. So each chromosome has to be replicated. So that's the point of that G2 and M checkpoint, is to make sure that DNA replication has proceeded, that there's no problems with the DNA, everything's been replicated, everything has been done. That G1 checkpoint is also checking for integrity of DNA, but it's checking to make sure that there's no major problems, that nothing has been lost or left behind during the separation of DNA into daughter cells during mitosis. So back in chapter 6, we talked about the structure of DNA. There were two men who were really important in elucidating the structure. Their names were James Watson and Francis Crick. And they published a really short paper describing the structure of DNA with the sugar phosphate backbone and the nitrogenous bases in the center. But the most important feature of their structure of DNA as we know it today is the idea of complementary base pairing. And one thing they said when they wrote their paper is that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Basically, what they were saying is that because of complementary base pairing, each strand of DNA can act as a template for the other. So this process of DNA replication occurs when the parent molecule separates down the middle. So it basically unzips. Remember that these base pairs, these complementary base pairs, are held together by hydrogen bonding. Now, hydrogen bonds are individually weak but collectively strong. So that means that those bonds work to hold the DNA molecule together. But as you start to unzip, you just unzip small bonds at one time. So eventually, this parent DNA molecule is separated into two strands. Then complementary base pairs are added by an enzyme called DNA Polymerase all along that strand on both sides. Now these outside strands are our original parent strands, and these inside strands are the identical copies, the complementary copies, I should say complementary, the complementary copies of the individual parent strands. But you'll notice that it produces identical strands from those complements. So this idea of complementarity is really, really important in the process of not only gene expression that we saw in chapter 6 with our messenger RNA and our tRNA reading the mRNA code, but also in this process of DNA replication, because it allows the cell to always know what the other strand is and to make a copy of the other strand in a faithful manner. So let's review a little bit about DNA structure. Remember that we have this 5-carbon sugar. Deoxyribose is our sugar. The reason it's called deoxy is that at this carbon labeled 2', this 2' carbon is actually lacking a OH molecule compared to the same position in RNA. So there are two major differences between DNA and RNA. One is the sugar. In DNA, this sugar is deoxyribose, and in RNA, it's ribose because it has that oxygen. Remember that sugar is attached to a phosphate group. So we've got a phosphate and a whole bunch of oxygens up here. Remember that makes our DNA molecule overall negatively charged, which allows us to run it on an electrophoresis gel. And then recall that in the middle we have our nitrogenous bases. These are our single ring pyrimidines and our double ring purines, and they bind complementarily with the opposite. So A binds with T with 2 hydrogen bonds, G binds with C with 3 hydrogen bonds. Now, these labeled carbons, these prime carbons, are actually important for a particular reason. And the reason is that they are labeled to show us that DNA actually has a direction. Now, they're also labeled because that's what you do in chemistry. You label your carbons. But it's important here, not because we label carbons, but because it actually shows us that DNA has a direction. Now, if you look here, we've got this 3' carbon here and this 5' carbon here that is bound to this phosphate group. If you look over here, we have our 3' end, and we can add on a new molecule with the 5' end. So we always attach a 3' end or the 5' end onto a free 3' end. So one end of our molecule is going to have a free 3' end. The other one, if you followed it all the way up to the top, would have a free 5' end. On the opposite strand, going the other direction, we have a free 5' end here, and if we were to look at the other end of this strand, we would be adding nucleotides to the 3' end way up on the other side. So in the process of DNA replication, we can actually only quote-unquote grow DNA in one direction. We always have to add our nucleotides to the free 3' end, and we connect them to the phosphate on the 5' sugar. So this is how we get our sugar phosphate backbone. We have our 3' sugar attached to our 5' phosphate, attached to our 3' sugar, attached to the 5' phosphate. All right, so let's look a little bit more closely at the process of DNA replication. Now, the process of DNA replication uses the idea of complementary base pairing. And when it does that, it has to make sure that it is maintaining the integrity of information. So remember, that is the two key features of DNA replication, that it uses complementary base pairing and that it faithfully replicates. In other words, faithfully puts in the correct nucleotide with its complement. So there are two major steps in DNA replication. In the first step, an enzyme here called DNA helicase actually opens up the double-stranded DNA and unwinds it. So A's signifies that it's an enzyme. So this is a protein doing a job. Helix means the double helix. So it's doing a job to open up the DNA double helix. So that enzyme binds to the DNA and moves along, pulling the strands apart. This region here is called the replication fork. I'll let you use your imagination as to why that's called a fork. And you'll notice that over here we have our strands separated. And if you recall from the binary fission picture in the first video, this is where we had those light blue strands sort of adding complementary base pairs to the parent molecule. So unwinding and separation, we've got our helicase unwinding the double helix and separating our strands, releasing those hydrogen bonds between those base pairs so that the strands can open up. After that, we basically get our copying. So at this point, we have another really important molecule called DNA polymerase. So DNA polymerase is going to bind to the molecule. It actually would have bound down here. It's going to bind and then it's going to move along basically like a train along a track, except this train is building the track as it goes using the template from the other parent. So recall from the last slide that I said that we add complementary base pairs to the 3' end. So DNA polymerase will bring in a complementary base pair and link the 5' end of that nucleotide to the free 3' end. Now I'm going to give you a little hint here. When I ask you on a quiz or on an exam where the nucleotide is added, you are going to say, hopefully you said to the 3' end. That's what you were supposed to say. So when that question gets asked, you are going to tell me that the nucleotide is always added to the 3' end. This is why we say that DNA runs in the direction of 5' to 3'. So the elongation phase is the phase in which DNA polymerase moves along the DNA molecule and adds the complementary base pair to the parent strand, making a new copy. Now this happens on both sides of the DNA strand, the DNA double helix. But the other strand actually occurs in the other direction. Now this is because the 5', 3' end runs the opposite way. So here our DNA polymerase has bound and it is adding nucleotides in the 5' to 3' direction, always adding the next nucleotide at the 3' end. So adding it on here, chugging along the track, eventually it's going to meet up with this other DNA polymerase and the pieces are going to be bound together and we'll have a new string. As the DNA double helix moves on, in this case moves up, this DNA polymerase is going to continue along the track. Another one is going to come in here at this end and move 5' to 3' in this direction. So this happens at the same time. Both strands are replicated at the same time. And the replication happens until the entire strand is copied, in which case you get two identical strands that were made from the original parent strand. So one of DNA polymerase's jobs is actually to perform proofreading and error correction. So this is just like when you give your paper, a paper that you're writing, say for English, to your best friend or your mom or your girlfriend or whoever, and ask them to proofread it. So what are they doing? They're looking for mistakes so that you can fix them. That's exactly what DNA polymerase does. It does this by recognizing mispaired bases, misbonded bases, because those errors cause structural problems in the DNA molecule, and DNA polymerase recognizes that. They can take out the erroneous base pair and put the correct one in. So DNA replication is actually pretty good. It doesn't make a lot of errors. Only one error in about 10 billion, 10 million, 10 million base pairs. Trying to recall my stat there. So not a lot of errors. But that doesn't mean that those errors don't mean anything. Remember that in an earlier chapter, I believe it was maybe chapter 4, we said that there's non-coding regions of our DNA. So if an error occurs there, it's not going to make any difference really. But if an error occurs in a coding gene, it could cause a lot of problems. So in general, errors and mutations are going to be a bad thing. The reason that they're a bad thing is that organisms are kind of already good enough. They already have the genes that are helping them to survive in their given situation. And if any gene changes, it's very likely to put that organism at a disadvantage. If a protein stops working, if you get a loss of function due to a missense or a nonsense mutation, the protein's not going to function, the cell might not function, and that could have drastic implications for the organism. But sometimes those changes are a good thing, or even a neutral thing. Sometimes you just get a change where maybe color changes a little bit and you go from dark purple to light purple, or brown eyes to blue eyes, something that doesn't really give you any particular advantage. Those are neutral mutations, or sometimes a change occurs that really makes no difference at all. Other times, every once in a while, a mutation will occur that actually gives the organism a new protein or a new function that's actually advantageous. So we often think of mutations and errors as bad, but every once in a while they're actually a good thing. And they are a source of new genetic information that evolution can act on. So when you get changes in your DNA, evolution begins working on those changed traits. And sometimes they're advantageous, other times they're not.